Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Armor CAR T Cells with Enhanced Potency for Cancer Immunotherapy. I'm Charlotte Barker, an editor at BioInsights, and joining me today is Saba Ghassemi, who will explore how maintaining a stem-like, non-activated CAR T cell phenotype and using metabolic and cytokine engineering strategies can enhance CAR T persistence, function, and long-term anti-tumor efficacy. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to Saba using the Ask a Question box at the bottom of your screen, and we'll try to get through as many of those as we can during the session. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now I'll just briefly introduce our presenter. Dr. Saba Ghassemi is a leading researcher at the University of Pennsylvania, where she develops potent, rapidly manufactured CAR T cells through innovative engineering and immunology approaches to enhance their clinical efficacy and accessibility. So without further ado, I'm going to hand over to Saba to kick us off with a presentation.
Thank you very much for the introduction. I would like to talk about the advances that we made over the past couple of years on the non-activated CAR T cell platform. So as many people are familiar with this slideshow, this is an overall process of generating CAR T cells for adoptive immunotherapy, a process that T cells are isolated from the patient and cultured ex vivo over typically 9 to 15 days in a very nutrition-rich environment, and they're then genetically modified with a CAR. This is the area that I'm interested, the highlighted area, and the overall goal is to increase the potency of our product, the CAR T cells. And we have good reason that the process that we have right now is suboptimal. The success of CAR therapy depends on several parameters that they generally intersect with each other. For example, if we use different co-stimulatory domain for the CAR design that we're using, that usually impact the persistence of CAR T cells. What we are primarily interested in is improving the quality of our product, CAR T cells, and in general, when I mention the quality, what I mean is the differentiation state of these CAR T cells or their metabolic fitness. We recently showed that activated T cells progressively differentiated over time. So by the end of their expansion time, on here we showed, as you can see in the panel on the right, on day 9, they accumulate more effector differentiated progeny, which lacks quality, but and they do not persist. So they're great killers, but they don't persist. And they're because of that, they're less effective for T cell-based therapies. You can see on the panel on the left some of the important subsets, such as naive like T cells or stem cell memory. After several days of activation and expansion over time, we completely lose that subset. And at this point, there are many studies have shown the correlation between the subsets in the product that we infuse to the patients that correlates with the success of CAR therapy. For example, in a NAM6 mouse model of leukemia, we previously showed that after we established the xenograft in the mice, we infuse cells that are expanded for shorter time. Here we have for 3 days, and a medium time like 5 days and 9 days. As you can see in the graph in the C panel, the day 3 cells, they outperform day 9 cells in anti-leukemic activity. For this experiment, we use a subtherapeutic dose, so we want to stress the system so that they don't cure. But you could see of the BLI measurement, the cells that are expanded only for 3 days, they can clear the tumor, whereas cells that are expanded for 9 days, they completely lose the control of the tumor. And also the data of the number of T cells in the peripheral blood also confirm what we're seeing, as in the group that are expanded only for 3 days, we can see a lot more T cells in the blood compared to the day 9 cells, which this number of T cells, they correlate with the persistence of T cells over time in the blood. Founded on this insight, a clinical trial started at Penn to test the efficacy of a 3-day product versus a standard 9-day product. This was the first in-human study that evaluated the humanized CART19 that also are further modified to secrete the pro-inflammatory cytokine interleukin IL-18, basically. And based on the clinical data that I show here, and this study is already concluded with 81% complete response. These highlight the potency of these cells compared to the 9 days expanded process. And what was very important about the study was the dose that we used compared to the standard Kymriah process that was FDA approved a few years ago at Penn. And so for this process, the dose we used between 3 to 7 million CAR-positive T cells, where the standard dose for the day 9 process is 600 to 800 million. So there's a point here that I think is very important for us to know, that the number, the dose doesn't matter as much as the quality of the T cell. So technically, if you have a lower dose of really high quality T cell, that could be enough compared to having a large dose of 600 million to 800 million of mediocre CAR T cells. So it's very important for using the novel platform that are coming out to shorten the manufacturing process. So based on those data, we hypothesized that if we eliminate the TCR activation by using the anti-CD3 and CD28 coded beads, if we can effectively do the gene transfer to the T cells, that process, if we eliminate the activation process, would yield a T cell product with high functional potency. And that means that, we hypothesize that, we can generate CAR T cells that are made of less differentiated phenotype T cells. And that, when we adaptively transfer these undifferentiated or less differentiated phenotype CAR T cells, we would have better and in vivo functional activity. So here we use the same NAM6 mouse model of leukemia. For this platform, we used 2 different processes. Either our standard process of day 9 activated and expanded over time. So here, we get the cells, we activate with the beads, and then we add the lentivirus, and over time, we expand them about 9 to 10 days. And at the end of the study, we can wash and, freeze or, inject to the mice. And we compared this with the other platform, the non-activated platform. In this platform, what we do is that we get the cells and then we just mix it with lentivirus, and after 24 hours we can wash and inject to the mice. And here we used a stress test, in vivo functional stress test, using limited number of non-activated CAR T cells, and compared it with the standard model. So if you can look at this graph, on the day 9, the standard basically platforms, they can clear the tumor very quickly, but all of our mice relapse here. But in contrast, if you can look at the non-activated groups, that they have different doses, there is a sustained control of NAM6 growth that is associated with the persistence and engraftment of CAR T cells, as you can see on the bottom graphs. This gave us thought that, okay, now based on the data that we have, can we just extend this the humanized card19, extend our clinical trial and use non-activated platform. So here we have two products. We have humanized card19 either with GFP control or IL-18. And here in collaboration with Axion BioSystems, we used the impedance platform, which is Maestro Z impedance system, to look at the real-time output of cytotoxicity of these cells. So here, this device, they can create impedance. After we add the tumor cells, they can create an impedance, and over time, after we add our effector T cells, which is here our non-activated card19 GFP or non-activated card19 IL-18, after they start to kill the target cells, they impact on the impedance signal. So one of the benefits of the system is that it enables the real-time monitoring of killing dynamics, as opposed to the general chromium-based killing assay that we had the output like at the end point, and also it's not very easy to use the chromium-based, and so it often gives us a valuable insight into potency and kinetic of the CAR-T performances. As you can see here in the graph, we're either have tumor alone or NTD, that you could see like the impedance signal doesn't change. It increases over time because they start to grow, and but here after we add the effector cells, the impedance signals start to diminish and with a different rate. So the red line belongs to card19 IL-18 non-activated, and then the blue line is card19 with GFP. Then we thought to test it in a mouse model. Here we use a GECO1 mouse model, and we compared, we used the same product that we used in the clinical trial, a 3-day manufactured activated card19 IL-18, and we also, as a control, we included activated card19 alone as well. And we compared it with a non-activated system. On the upper graph, you can see either with or without IL-18. And here you could see the real winner is non-activated card19 IL-18. They can clear the tumor so well, and they can persist over time. And I have to mention that for all this experiment, we use a really low dose. Try to get close to subtherapeutic dose so that we can distinguish which of our group can outperform the others. In order to remove the effect of the time, because in the previous slide, one of the group was 3-day activated, one of them was 1-day non-activated. So here we compared two groups that are either they're both expanded for 1 day, either in activated system or non-activated system. So we have the humanized card19 IL-18 and compared it with, in one group we activated, in the other group we did not activate it. We only exposed them to lentivirus and compared their performance. And as you can see, the red line belongs to the non-activated card19 IL-18 and blue one is activated 1 day card19 IL-18, and they can both clear the tumor with non-activated humanized card19 IL-18. They outperform all the groups. And if we look at the number of the T cells in the blood, we can see that by day 38 for the non-activated group we have almost a log difference in the number of the T cells in the periphery of the blood, which that represent the persistence of these T cells. We also wanted to test their efficacy in a solid tumor environment. So here we used a pancreatic cancer xenograft model, and we used mesoCAR either with GFP or IL-18 as a control. So we have meso IL-18 versus meso GFP, and we compared the efficacy of these CAR T cells in this xenograft model, and used several doses to compare the doses. But the real endpoint here for us was to make sure to see if these cells, they have the non-activated platform, they have also efficacy in solid tumor environment. And as you can see here, they can clear the tumor, and it's very dose dependent, of course. For example, the red line is meso alone, and then the green line and the brown line is meso CAR with IL-18 with different doses. As we decrease the dose, it takes longer for them to clear the tumor. However, all of them, they can clear the tumor, which shows the quality of these CAR T cells in terms of the tumor clearances. We redid this. We just wanted to see how long we can, are the non-activated, they can persist in the solid tumor environment. And as you can see here, we kept these mice up to 90 days. And the meso CAR IL-18, they can clear the tumor a bit faster compared to the meso alone. They're both non-activated, but I think the point here that I want to mention is that these CAR, the non-activated platform, the non-activated meso CAR, whether with IL-18 or not IL-18, or in solid tumor, or in a blood-based tumor xenograft, they are very potent. So it's very feasible for us to generate CAR T cells within 24 hours that they are non-activated. They're extremely potent and they're durable. They have this durable anti-tumor function that they can persist over a long time. So this shows the benefit of this platform, and compared to the conventional platform that is used right now, that activate T cells and expand over 2 weeks, right? And again, that shows that that's not needed, because what we do is that we over time we lose the quality of CAR T cells. Another experiment that we've done, we just wanted to make sure that we can use patient samples to generate the non-activated CAR T cells. So here we used patient apheresis samples that we have, and from that we used using non-activated platform, we made either CAR-19 GFP or CAR-19 IL-18, and we expanded over time. And as you can see, they can perfectly clear the tumor. And if we look at the number of the T cells in the periphery of our mouse blood, we could see that the group CAR-19 IL-18 has a log more, not many logs more T cells compared to the CAR-19 alone. The CAR-19 that our control CAR-19 group that is co-expressing GFP. That shows the benefit of including the IL-18, and that gives us confidence to move on with the planned clinical trial that we have to extend the CAR-19 IL-18 that we had on 3-day manufacturing. Now we're planning to have that in a 1-day non-activated CAR T platform. And this in general, I think all the data that I show, I would like to convince people that we can get closer to the point of care solution or point of region solution for the CAR T therapy. If we can, use such platform that, here, we use non-activated 1-day CAR T, we can completely eliminate all this process that we have that is very cumbersome, and use a lot of maybe material or that is not accessible, such as let's say human serum. At some point we're going to use all the human serum available out there, or the number of users that we need to expand these T cells for 2 weeks. It's very costly, and we can introduce a lot of errors. Also, if you think about it, if you have a one-day process, it's a lot easy to automate the system compared to have automation on the 2 weeks process, because this is another active area that many people are trying to automate the system, because in this case we can have higher accessibility to more patients. So for conclusion, I like to convince everyone that we are able to generate CAR T cells within 24 hours and non-activated CAR T cells. And these cells are extremely potent and they are durable, and their persistence is a lot higher than activated CAR T cells. And the other point that I would like to mention is that the cytolysis data that generated using the Maestro Z from Axion BioSystems, we can say that it closely correlates with our in vivo outcome. So it's a great system, because what I like about it is that it gives us information in real time, so we can see which CAR T cells can kill faster compared to the other groups. And also another point that I like to mention is that very low doses of CAR T cells are sufficient, providing that they are high quality. So quality matters compared to the quantity. So we can work with a small number of high quality T cells compared to the large number of low quality CAR T cells. And of course, using such platform has implication in terms of cost, in terms of automation and accessibility to more patients, and more importantly for patients that they have progressive disease and they cannot wait until the whole process of manufacturing CAR T cells for a long time. And unfortunately, many times we lose patients in this process. So I think it's very important to develop a process that we can provide such a great therapy faster to more patients. And with that, I would like to thank my collaborators. And of course, we're a true believer of teamwork makes the dream work. So we have so many people always working with each other, and also the funding and the collaboration with Axion BioSystems, that they've been great in terms of instrumentation, and also providing all the feedback in our experimental design.
Perfect. Thank you very much, Saba. It's a great presentation. We're getting lots and lots of questions in about it. So we'll move into the Q&A now. So let me see. First question that I have here. Could you describe the rationale for IL-18 armor strategy? And if IL-18 is expressed, would you define a T cell that expresses IL-18 as activated?
Can you repeat the second part of the question after and if?
And if IL-18 is expressed, would you define a T cell that expresses IL-18 as activated?
Well, the rationale to use IL-18, it's part of a maybe more general rationale that we have to try to enhance the quality of our product, right? So in this case, we try many ways, whether their manufacturing process, whether their quality, whether we can enhance by different genes, genetically engineering them. And in this case, IL-18 is a pro-inflammatory cytokine, and there has been many studies that they show that if we co-express IL-18, CAR T cells, they have a better function because they can produce interferon gamma, and we can increase the function. So in the same line of trying to up the defense of our CAR T cells from the tumors that they have in front of them. So that's what they have. So and if they are activated, well, they are activated via their CAR. With the target, through in vivo, not TCR.
Right. Brilliant, that makes sense. And next question I've got here, for one-day manufacturing using lentiviral vector, do you formulate the dose based on transduced T cells or total T cells? And what's your transduction efficiency of 1 day versus 9 day?
Yeah, so great question. Within the 24 hours, with the current system, which is technically flow cytometer, we cannot see the CAR positivity. So for our in vivo experiment, generally we go with total cells, but we've done studies using PCR-based methods to estimate the number of CAR positivity within 24 hours. And we have a very general, like 10% CAR positivity. I have to mention that we've been very active in terms of increasing this number, and which we've been successful. And what we do now, we correlate later on the amount of CAR positive that we get from T cells that we get from the peripheral blood, and we have a better estimate right now. But we go with total T cells.
Perfect, thank you. And do you use cytokines in your one-day non-activated T cell manufacturing?
Yes, we do. We use IL-7 and IL-15 because they kind of make the T cells permissive for transduction, because these are non-activated and they need a signal in order for the endocytosis happen, and so on and so forth. So we use IL-7 and IL-15.
Great. Brilliant. Lots of questions coming in. Do keep them coming in. Any we don't get to today, we'll answer by email. So keep them coming in and we'll get back to you one way or another. Next question I've got here. Can the Maestro Z assay be used to model hypoxic conditions like those found in the tumor microenvironment? And have you tried looking at CAR T cell function in hypoxia using this platform?
Yes, absolutely. It's actually brilliant because we have metabolically engineered CAR T cells with different genes that they can overcome the hypoxic condition. So we've had great experience with using Maestro Z in hypoxic condition, because we can see our engineered CAR T cells, they can overcome hypoxic condition compared to our standard CAR. And I'm hoping that I can present that data very soon.
Brilliant, thank you. Is the vector amount and/or the starting cell number the same in non-activated versus activated T cell protocols, or do you use more lentiviral vector for non-activated?
Generally, we use higher MOI of lentivirus for non-activated versus activated. However, again, as I mentioned, we've been very active in terms of increasing the transduction efficiency using many different simple ways for non-activated, trying to balance that, trying to really kind of like use the same MOI that we use for activated compared to the non-activated, using these methods. And yeah.
Brilliant. Now, a question here relating to a specific slide. So we can't bring the slides back up, I'm afraid, but hopefully you'll remember what we talk about here. So what happened to ACT-free meso IL-18, the yellow line on slide 14?
I think I think we had toxicity, IL-18 toxicity. I assume we didn't run any official toxicity experiment to just make sure is that, but that's my guess. But for some reason our activated just died. But it just we've repeated the experiment and we didn't see that. It's not something that we see often, but my guess is that there's some sort of toxicity happened there.
Interesting. And how well does the in vitro data from Maestro Z match with what you see in your mouse studies?
It's very, they correlate. It's very insightful. I think one of the benefit of Maestro Z is that we can see in real time what's happening compared to checking, like, let's say, in a standard cytotoxicity assay that we check on 24 hours or 48 hours. You can see which group can respond faster and with a higher speed to when we add the CAR T cells. So that's, I think, very insightful, and they correlate as we see.
Perfect. Well, we're almost out of time. Let's just squeeze one more question in. Have you seen a trade-off between initial potency and long-term persistence when comparing activated versus non-activated CAR T cells? If you can answer that briefly, that'd be great.
No, I mean, part of the potency is the persistence, and non-activated CAR T, they're very potent. They are potent. They have sometimes a lag just to start, but that's like kind of playing with the number, CAR T number, but they are very potent. And in cases, if we saw like in the 1 day activated versus non-activated, they're even faster than activated. So there is no trade-off.
Perfect, thank you. Okay, great. Thank you very much, Saba, for answering those questions. That is all we've got time for today, as today was a live 30th. Any questions we didn't get to, we will reply by email though. So don't worry, we will get to you. The webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with some feedback on today's webinar, and a link to that can be found in the resources on the right-hand side of the screen, or that will also be in the on-demand email. There's also an attendance certificate available. That's also on the right-hand side of your screen. So all that's left then is to thank Saba once more for a great presentation, and thank you very much to the audience for joining us today. We do hope you'll join us again soon.